This technique is a dependable way to deliver any content, such as drugs, proteins, or cells, to specific locations in tissues or animals. You can track the delivery and tune the release of the contents to fit the experiment you desire. It can be used to treat numerous diseases from wound healing to ocular and cardiac diseases.
The main advantage of this method is the ability to deliver therapeutic cells using alginate microcapsules to the target sites without causing any immune reactions, and maintain a sustained delivery of a fresh secreted therapeutics for a longer period of time.
Begin by culturing retinal epithelial cells according to manuscript directions. Passage the cells when they reach 70 to 80% confluency using standard tissue culture procedures. To encapsulate the cells, mix sodium alginate with deionized water for a final concentration of 2% weight to volume. And purify the mixture via filtration with a 0.2 micrometer sterile syringe filter. Trypsinize, centrifuge, and wash the cells with 10 millimolar Heapes buffered saline solution. Use a hemocytometer to count the cells and adjust the final cell concentration to 1 million per 1 milliliter of alginate solution. Load 300 microliter aliquots of the alginate and cells mixture into a 3 milliliter syringe and attach it to a syringe pump. Prepare a gelling bath according to manuscript directions and place it in a sterile 50 milliliter beaker below the syringe tip at a 7 millimeter needle to bath.